Yeah, we are partially opening up PhD students, starting with PhD students.
Okay.
And maybe by October, we are planning to bring some BSMS students also partially on a phased out manner. Yeah.
Good. Yeah, slowly, it's good to bring them, check it out and see how it goes.
Yeah.
Yeah, COVID is here to stay. We have to live with COVID.
Yeah, we have to sort of plan. It's like flu. We have to start living.
Exactly. We have no way.
I don't know how we are going to survive this variants, though.
I know. 78% of rural India is unvaccinated, and it's not going to get vaccinated soon because there's just not enough vaccines going around. So,
and I saw yesterday that the Lambda variant might be even worse.
There we go. Hi, Ralph. Good to see you.
Hi, thank you. Good to see you again.
Yeah, it's been a while. You have not been with us for a while, so welcome back.
Thank you.
Hi, Alex.
Hi, Surajit. Nice to see you again.
Yeah, so you're traveling a lot, eh?
Yes.
Good.
I traveled, now I'm back to base.
You are a brave man.
Well, I'm fully vaccinated and I try to observe all the precautions. So.
Yeah, might as well. My bedroom has become too comfortable lately. Not been getting out at all. All right, it's about 11 o'clock. So, I have to introduce you, Hema. All right, we are almost ready. Let's see, do we have all the bodies more or less? Some missing. There we go. Sujay has come. Hema and Sunny, the old timers at Buffalo would know. Okay, so let's get started. It's a real pleasure to introduce Professor Hemachandra Subramanian. And he'll speak on structure-function relationship in DNA, the importance of kinetics. Just to briefly introduce him, Hema graduated from SUNY Buffalo with a PhD in theoretical and computational condensed matter physics in 2012. And he did his work under the supervision of Professor Jong Han, my colleague. And he worked on correlated systems and stuff like that. He then joined the Moffitt Cancer Center, quite a U-turn, and Research Institute in Tampa, Florida as a postdoc, where he began exploring the origins of information acquisition and storage in protobiological systems. Hema worked in the group of integrated mathematical oncology, headed by Robert Gattenby. He moved to National Institute of Technology, Durgapur, in 2018, joining the physics department as an assistant professor. He's currently setting up his research group and continuing his research on the evolutionary significance of structural properties of DNA. I know that he is very happy to actually be funded now. And Hema actually was always a very creative person. He always asked difficult questions. So whenever I gave talks in Buffalo, I remember I used to watch out for this guy because you never know what he's going to throw at you. So this is my turn to take revenge. But at any rate, the floor is yours, and thank you so much. And we'll mute ourselves such that you are the speaker and we can always ask questions as we go along. Thank you.
Thank you, Surajita. Thank you so much.
You can jack up your volume a little bit, maybe.
Okay.
Or maybe it's okay. It's okay. Don't worry about it. It's fine.
Okay. Thank you so much, and I'm really honored to talk in this lecture series with the lineup of people, very distinguished people. So thanks for giving me this opportunity, and I hope I do justice to the to your confidence in me. Is my screen visible?
Yes. Do you want to put it in presentation mode or keep it like
sure,
this?
Sure. Yeah. Is it okay now? Can you see the screen?
Yeah, that's good.
Okay. I'm glad to be giving this talk to this close-knit group of people. Of course, my teachers are here. My former teachers, Professor Panigrhi is here. Professor Surajit Sen is here, and possibly Professor Han as well. He said he will join, but I'm not sure. And some of my colleagues, some of my mates, classmates are here. Sunny is here. So I'm glad to be talking in a forum like this. And so I will be talking about the stuff that I've been doing for the last 9 years. So we start off asking a question that was too heady at that time. How is information stored or processed in biological systems? How do biological systems acquire information and store and process information? A rather big question, but we wanted to start simple. So I started playing around with modeling autochiraltic reactions, reactions where heteropolymers make a copy of themselves. Initially, when we try to play around with the kinetics and thermodynamics of these heteropolymers, luckily we found something very interesting, and that was that we found a hint of symmetry breaking. I'll tell you about how that happens, and everything started off from there. This whole DNA structure function relationship and the kinetics and everything started off from there. So let me explain what we, so obviously when we start off with research, we weren't trying to answer the questions that this talk is going to address. We were trying to answer something else, completely different. Those questions were equally deep, but they were not related to anything we eventually ended up answering. I hope we answered the questions, but maybe it is not, it's wrong, I don't know. We will see as experiments come in. Okay, so to give you an overview of DNA replication, before we start off with heteropolymers, before we go general, we will talk a little bit about how replication happens, self-replication happens in extant systems, current DNA systems. You can see that there are two strands, the double-stranded DNA. There are two strands. There's 1, 3, 5 going from left to right, and there is a 5, 3 going from, again, left to right. So what happens is when a DNA wants to replicate, it creates a little bubble in the middle. You can see that in the red there. So it creates a little bubble, and then some RNA molecules come and attach to the bubble on each side, and that is called priming. So once it's primed, polymerases come in and start extending using DNA nucleotides, AGTC. Okay, and the bubble gets expanded on either side. There are molecular machines called helicases that unwind on either side. So these are powered by ATP. So they progress through the DNA. They unzip the DNA, making the double strands into 2 single strands, and the polymerization of the replica strand happens on both the strands, on the bottom strand and on the top strand. But there's a complication here, which is that the unzipping, let's take the fork on the right side. If you look at the fork on the right side, the unzipping happens towards the right, and the bottom strand is being built continuously in the same direction as the unzipping is happening. But on the top strand, the construction has to go from 3 to 5. So it's a unidirectional construction. The construction always goes from 3 to 5. The replica strand is always constructed from 5 to 3, which means the template strand is 3 to 5. So you cannot construct the top strand. You cannot construct the daughter strand for the top strand template continuously. It has to be discontinuous. You have to open up a little bit of DNA, single-stranded, make it a single-stranded DNA. Once enough DNA is available, once enough single-stranded DNA is available, you start filling that up with your daughter strand. Okay, so you replicate in fragments, okay, in bits and pieces, and then you splice them together. You concatenate them together. This is incredibly complicated. If you look at the machinery that is required to do this, there's a lot of proteins involved. And also it is error prone. There is a possibility of errors, misincorporations, SNP single nucleotide polymorphisms, and all that possibility opens up because of this discontinuous replication. The question is why? Why would DNA go to such length to maintain such a replicative organization? This looks very complicated. So why not just 2 parallel strands, let's say 3, 5 and 3, 5, and replicate both in the same direction? The templates are built in the same direction. So you don't have to go through this, this complicated bits and pieces replication. Okay. We will see why this is advantageous. Okay. So the question is, why are, to phrase it another way, this kind of replicative mechanism arises because there is a constraint of growing the daughter strand in only one direction. So if you look at the diagram again, the daughter strand always goes from the 3 end of the template towards the 5 end of the template. It always grows in one direction. It cannot grow in the other direction. There is an unidirectionality of growth. So if you do away with that unidirectionality, you can do away with this bits and pieces kind of replication. You can do away with this Okazaki fragments and replicate the whole, both the strands continuously. So the other way to ask the question is that why unidirectional replication? Why should replication happen in only one direction? Why cannot the template strand be extended, the daughter strand be extended backwards as well? So these are 2 related questions. Why is there unidirectionality of replication? And why is DNA anti-parallel? Both of these things contribute to this bits and pieces kind of replication mechanism. At the outset, let me make it clear that usual answer to this from chemists is that the DNA molecule dictates it. So it's the property of it's a molecular property, the structural property of the DNA molecules of AGTC nucleotides. But my question is a little deeper than that. My question is, why did evolution choose those nucleotides in the first place? Of course, DNA is a product of molecular evolution. It's perhaps one of the most evolutionarily selected, highly selected molecule. It's a central molecule of biology. So it should have gone through a tremendous amount of selection. So why did evolution choose such a molecule if it were so complicated, right? So our point is that there is some evolutionary advantage to this organization. The goal is to find what. Okay, so this is where we started, and the previous ones were added later on. But so we started out with modeling self-replicating heteropolymers, initially to understand how information is acquired by these proto-organisms, self-replicating heteropolymers, basically. and eventually ended up answering some of those questions that I asked before. So our goal is to maximize the replicative potential of these self-replicating heteropolymers. What kind of properties would you would you give to the molecule to make its self-replication superior, to make it evolutionarily more successful? What kind of properties are needed for the molecule to replicate faster? Okay, and that's the question. So how would you make them? Basically, it's in silico evolution for self-replicating heteropolymers. Okay. So what we tried to do was to assume a template, a heteropolymer template, or a polymer template. Just for now, it's just a polymer, and you want to have you want to create another daughter strand on top of the template strand. How would you do that? Let me, if you can see this. So basically, I'm just going to draw. So you have this template strand, and you will have a lot of free-floating monomers around, and they would come and sit on top of this this template strand, and they're going to polymerize eventually. Okay? And that's the goal. That's our goal. Well, okay. Okay, so that is, do you see the video? I think the video is visible now. I am.
Yeah.
This is, it's just a, it is not the, this is not the simulation. It is just a cute picture. Okay, this has nothing to do with the numerical simulation, but it's just an illustration to show what the simulation would do. Okay, so you can see that the monomers coming and binding to the template. Okay, rather haphazardly. There is no. They're not continuously. They're not attaching to the template in an orderly fashion. Okay, so how would you polymerize when two hydrogen molecules are located on a different parts of the template strand? How would you polymerize that? So what you want to do is to have two molecules, two nucleotides sit right next to each other. And how would you do that? You would let one of the H bonds, one of the hydrogen bonds, catalyze the formation of another hydrogen bond right next to it. So if you do that, you will have each hydrogen bond reduce the kinetic barrier for the next hydrogen bond to form. Okay. So, okay. So you will have two hydrogen bonds. Let me again go back to the diagram. You will have this kind of a template, and you will have hydrogen bond coming in, and this hydrogen bond will catalyze the formation of the next hydrogen bond. So another monomer would come in, bond, and this would catalyze the next bond, and this would catalyze the next bond, and so on. Okay. So this way, you're not wasting your monomer. So another monomer is not going to come and sit here and form a bond which is useless. Okay, so because there is no way of polymerizing it. So here there is a way of polymerization. There is a way of covalent bond formation between these, so they get stabilized. Okay, when the nucleotide forms elsewhere, there is no stabilization. It's just wasted. The monomers are wasted. You don't want to have that. So you basically catalyze the neighboring bonds. By introducing this catalysis of the neighboring bonds, you bring a lot of these monomers right next to the growth front and not away from the growth front. All right, but there is a problem here. What you're doing is you're catalyzing the hydrogen bond, so you're reducing the kinetic barrier. So the hydrogen bond can form, and also it can break off because you're reducing the kinetic barrier. What you would want to have is once the hydrogen bond forms, once the monomer comes and sits on top of this template, you would want to have that bond stay. You don't want it to break. Okay. Remember, we are very close to the melting temperature of the hydrogen bond because you need to break and form the bonds. Form the bonds during polymerization and break the bonds for these two strands to separate again for the next round of replication. Okay, so you're basically very close to the hydrogen bond melting point, you're going around in cycle in temperature. So this is time and this is temperature. So you're going around in cycles somehow like that. And this is your TC. Okay. So polymerization happens here, and the strand separation happens about the TC. Okay. And both the strands are available for the next round of polymerization here. It's something like PCR, polymerase chain reaction, that happens ubiquitously everywhere. So your hydrogen bonds are forming very close to the melting temperature. So the probability of them bonding and dissociating is quite large. So you don't want, if you're decreasing the kinetic barrier, you're allowing the bond to form, but also you're increasing the dissociation as well. You don't want to have that. So you want to have bond formation, but not bond dissociation. What would you do? Okay, so the conflict, I'm saying the conflict here, writing about a conflict. You want to increase the kinetic barrier once the hydrogen bond is formed. You want to decrease the kinetic barrier for the hydrogen bond to form in the first place. Okay, so the solution, it turns out, is something called asymmetric cooperativity. Okay, you cannot play with the thermodynamics, by the way. If you increase the like something like stacking interaction between two hydrogen bonds, you're going to increase the melting temperature. Not useful. So you cannot play with thermodynamics. Your only leverage over this is kinetics. So you can play with kinetics. So what is asymmetric cooperativity? So for future videos, it's useful to look at this convention. So when the bonds are here shown as thick, then it says that it means that the barrier is quite high. When the bonds are medium, the barrier is medium, and when the bonds are really thin, the barrier is really low. Here it is difficult to bond and difficult to break because the kinetic barrier is really high. Here it is easy to bond, but also easy to break. Okay, and the influence of the neighboring hydrogen bonds. Once this hydrogen bond is here, it can catalyze the presence of the right hydrogen bond. Okay, when this hydrogen bond is here, you can see the bar behind it. It can inhibit, meaning that raise the kinetic barrier of the hydrogen bond formed behind it. Okay, so this is asymmetric cooperativity. The one in front of you, you're reducing the kinetic barrier. The one behind you, the hydrogen bond behind you, you're increasing the kinetic barrier. So this seems to help in satisfying both the constraints, in bringing the hydrogen, bringing monomers in for polymerization, and also keeping them on the template for covalent bond formation. Okay, so here is symmetric cooperativity, which means both left and right is equally catalyzed. Okay, you can see that the bonds change shape once the hydrogen bond forms. Let's say that's the leftmost. You see the middle bond has reduced kinetic barrier. Again, the kinetic barrier is reduced on both sides. That's symmetric cooperativity. Okay, this does not help. This is not helpful at all. Okay, it brings in monomers, but it does not allow the monomers to stay hydrogen bonded. So you cannot have covalent bond. You don't have much time for covalent bond formation. Okay, so polymerization does not happen. The monomer just floats off. Okay. Now we can see asymmetric cooperativity. This is the crux of our argument. Now, as you can see, once the bond forms, like the leftmost bond, the right side, the middle bond is catalyzed, whereas when the bond forms on the right side, wait for it. Okay, the middle bond, now you can see the left side is inhibited and the right side is catalyzed. Okay, it's going too fast. Okay. Okay, I think you get the point. So yeah, right there. You can see that the left bond is inhibited and the right bond is catalyzed. Okay. So once you have asymmetric cooperativity, this is what happens. The leftmost bond catalyzes the next one, and this catalyzes the next one, and the third bond catalyzes the fourth bond catalyzes the fifth bond catalyzes the sixth. And the rest of the bonds have high barriers. You can see that because of the inhibition from the right. So this bond inhibits the one behind it. it raises the kinetic barrier. So this does not break away. And this bond inhibits the one behind it. This does not break away. So all these 5 monomers from left, they have high barriers. So they don't float away. They have higher lifetimes and they can easily form covalent bonds. Okay? So you can see that there is a directionality from left to right. Okay. So we have broken the symmetry, and because of the symmetry breaking, you satisfy both the constraints. Both are conflicting constraints. One, on one hand, you want a reduced kinetic barrier. On the other hand, you want a increased kinetic barrier. And if you break the symmetry, you satisfy both. Okay. And this means, oh, sorry. This means that your hydrogen bonds have or your monomers have a directionality. Okay. If you look at this again, this particular double strand can be unzipped from the right easily, but from the left, you can see that the leftmost bond is very high, has very high kinetic barrier. It cannot be unzipped easily from the left. It can be easily unzipped from the right. So which means your 4 signatures for unzipping the DNA, dsDNA from either side would be completely different. All right. So that's apparently what we see. This is a paper from 1997, and they see unzipping of DNA from both ends, from E1 and E2. Those are the experimental signatures from both ends, and the signatures are completely different. The unzipping signature is completely different. Right, we can create a Markov model. So what we did was to create a simple Markov model to prove our point, basically. We know what we want, basically. So you can really construct a simple model to show that. So we do see 2 maxima. Okay, these deep red points have higher replicative potential. Okay, these 2 maxima. So you see the splitting. You have the X and Y axis here are rate modulating factors. This is for the left, the hydrogen bond is increasing the kinetic barrier. Here it's 1.608, it's increasing the kinetic barrier for the left bond. Here it is increasing the kinetic barrier for the right bond. Okay, and this is 0.2 and 1.4, which means that the left side is inhibited and the right side is catalyzed. Over here, the right side is inhibited and the left side is catalyzed. Both are equally probable. So either one of these solutions are going to be taken up. Okay. So that's a proper symmetry breaking, and we ended up publishing it in 2018. So this is the first message, that asymmetric cooperativity is useful for self-replicating system. This seems to be very general enough. So quite possibly in other self-replicating systems, if you find it even, let's say in Europe or someplace, they could possibly have the same asymmetric cooperativity, which means symmetry broken nucleotides, where left and right are not the same. Okay. So structurally, what that means is that your nucleotides cannot be symmetric on left and right sides. Okay, they have to be asymmetric. And in biology, it means there's a 3' and a 5' in your nucleotides. And biologists term it as 3' and 5'. And this gives you the answer for the first question: why is there why is the construction of the daughter strand one direction, unidirectionally? Okay. It's possibly because your asymmetric cooperativity dictates itself. Okay. On one side, it is kinetically easier to construct your daughter strand, and it is kinetically harder to construct a strand in the other direction. Okay. And this is because it's easier to bring in monomers and keep them in place for polymerization. Okay. So we'll go on to the next question: why antiparallel strands? What is the reason for antiparallel strands? Okay. Now you can see that there is a directional unzipping. Okay. It unzips from the right towards the left. Okay. And you can see that these 2 strands are parallel. Okay. Parallel strand, it's easy to unzip. And when you want to construct, you can construct from the right end to the left end, or from the left end to the right end, sorry. So you can, either way, it doesn't matter. Okay. what matters is on both strands, you construct in the same direction. Okay. So why adopt antiparallel strands? Now, if you're antiparallel, what happens here is that you completely lose a directionality. There is no directionality because these monomers are symmetric, left-right symmetric. If I can draw, you have a monomer like that, and you have a monomer like this. Okay. Now, both left and right are the same. If you just rotate it by 180 degree, you get the same thing. Okay, you get the same thing. So there is no left-right asymmetry here. This is left-right symmetry. Right? So you cannot use this. You cannot use the same monomers on both bottom and top strands. Okay, if you want to introduce asymmetric cooperativity again in antiparallel strands, we will come to the point. We will come to the question of why antiparallel strands later. But if you want to incorporate asymmetric cooperativity in antiparallel strands, you have to go with two different monomers. Okay, you have to go with two different monomers. The blue and the orange. Okay. Once you break that symmetry, once you the top and the bottom monomers are different, then you can incorporate asymmetric cooperativity. It's not if you turn 180 degree, it's not the same anymore. The orange would come up to the top, so it's not the same anymore. So the heteromolecular base pairing actually reinstates asymmetric cooperativity. Okay. Now let's go to the reason of why antiparallel strands. If you just take a look at the center. Okay, let's go back and come back again. You can see in the middle, the bonds are weak, right in the middle. Okay, and so unzipping is very predictable. The central two bonds break first, and then the bond breaking progresses towards the left and towards the right, just like the picture that we showed in the first slide. Let me go back to the first slide, sorry. Yeah, here is a picture, just like the picture, there is a little bubble in the middle, and then the bubble expands, okay? And that is exactly what happens here. Okay, that is exactly what happens here. You have a bubble in the middle bonds break because their kinetic betas are weak, and then the bubble expands on both sides. Okay, and this configuration is dictated by the choice of the nucleotides here. I've made the top half, the left half of the top strand blue, and the right half of the top strand orange. Okay. By that choice, I made the bonds at the center weaker. Okay, so they can easily break and they form something called the origin of replication. So that's one of the ways how origins are defined. The places where the bonds are weak, so they break easily, they form little bubbles of single strand, and the single strand bubbles expand over time. All right, so is this just a nice picture, or is there any reality behind it? Now you can see that if you count the number of blue and the number of oranges, or the difference in blue and orange, something called a GC skew would come up. Okay, let me explain that again. So what you do is number of blues minus number of oranges on the top strand. Okay, so if you count the number of blues minus the number of oranges on the top strand, and make a cumulative diagram of that, you keep increasing the number of blues, keep increasing. You add the number of blues, so the number of blues keep increasing, and after that, after the origin, the number of blues keep decreasing because your number of oranges keep increasing. Okay, so there is an increase and then there is a decrease. Okay, there is a V kind of a cumulative skew would give you a V kind of a picture. Okay, so if you take pretty much any DNA of prokaryotes, and if you plot the same GC skew, this is the kind of picture you would see, and that is what you get. So you observe this GC skew in almost all genomes. Okay, in prokaryotic genomes there is just one V, and in eukaryotic genomes there are multiple Vs. Okay, multiple such GC skew diagrams, GC skew peaks. Okay, so what that means is that at the top here is your origin of replication. This is where the G predominant site switches to C predominant site. Okay, and this is where you would expect the origin to be, and that is exactly where you find the origin. And this has been going on for this is a paper from 1998. And these days, when people write software for finding origins of replication, this is precisely what they do. They calculate GC skew, they find where the GC skew switches sign or turns around like that. It goes from increasing to decreasing, and then label that as a origin of replication. So now we have a mechanistic reason for why that is so. Okay. The switch in GC skew is because the organization of your sequence is like that. Okay, you have majority G's on one side and majority C's on the other side, and because of that organization, you have a central part where the kinetic barriers are low. Okay, and they unzip first, making a little bubble, and that bubble expands. Okay, so. This GC skew is usually explained as a consequence of the directionality of replication. This is this skew is assumed to be evolved over millions of years or billions of years or whatever. But here we have inverted the cause and effect. Okay, so the current consensus is that the GC skews is is the effect of directionality of replication. But what we say is that it is also the cause of directionality. Okay? So because of GC skew, the replication machinery would travel in only one direction and not in another direction. So if my replication machinery starts in the middle, it moves only towards the right on the right-hand side and only towards the left on the left-hand side. It's easier to do so. In the other way, if it has to travel other way, then it has to break higher barriers. It has to break through higher kinetic barriers, not very favorable kinetically. Okay, so there are other experiments, single molecule experiments where people have measured incorporation kinetics. So when you have a G on the left, and what is the rate of incorporation of other molecules to the right of that and to the left of that. So you can see the asymmetry here. I don't want to belabor the point, but you can see the height of the blue and height of the blue on this side on G and C, they are inverted. Okay, so it's kind of neatly aligns with what we say. Okay, the left-hand side is inhibited. It's 1091. The right-hand side is catalyzed. This is 1256. And if you invert that, it inverts. Okay, so it kind of aligns with what our intuition says. Okay, let's switch gears and do I have time? Okay.
Yeah, you're doing well. It's 11:38 or so.
Okay. Now, if you look at this diagram, the top strand has only one flipped hydrogen bond. Basically, the blue is in the bottom and the orange is on the top. And because of this flipping, these two bonds have become stabilized. So unzipping, for unzipping, this creates a barrier. Okay, this is not going to unzip very cleanly. Okay, in a primordial scenario, you cannot have such a barrier because okay, I'm getting a call because there were no unzipping machineries available in the primordial time. So you have to unzip by yourself, which means these kind of barriers are going to make you completely lose out in the evolutionary game. Okay, so you need to have either all of the top strand nucleotides the same kind, or you lose out. Okay, so with just two nucleotides, I'm sorry, with just two nucleotides, this is what happens. Okay, your unzipping suffers when you try to store information. So either all the upper nucleotides are the same type, or you suffer unzipping. You suffer high kinetic barriers. Okay. So you cannot store any information in this system. There is no way of storing information. So what do? You go for two other monomers. Okay. You of course need two other monomers, not one, because excuse me. I need to put this on. Sorry. Hello. Put it out, please. DS6. Yeah. DS6A. DS6A. I'm sorry. Some delivery guy. He's looking for my house.
Real life.
yeah, I'm sorry. So now, if you introduce two other monomers, you can unzip continuously, and at the same time, you can store information. Okay, so effectively, you get four monomers. Okay, that's what you find. It sounds like conjectures, but it all aligns well. And again, the triplet genetic code. For the kinetic barrier of the middle bond, you need both the sides. You need both the left bond and the right hydrogen bond to uniquely determine the height of the middle bond, the kinetic barrier height of the middle bond. So you need to include all the three together if you are to use the kinetic barrier of the middle bond for storing information, right? So again, the central barrier, the central bond dictates polarity and solubility. That's the most important. The central nucleotide of a codon dictates what kind of amino acid, what type of amino acid is used in a genetic code. Okay, let's switch gears again and talk about chirality a little bit. Now, as I said, I'm sorry. Sorry. So your template, your daughter strand is constructed in one direction, okay? And the molecules, the nucleotides, have to have be asymmetric, 3 and 5. Okay, it has to have a certain orientation, right? So when a molecule has a certain orientation, when it forms a hydrogen bond, it has to come and form the hydrogen bond in the right orientation. If it forms in any other orientation, your polymerization is going to suffer. Okay, so you need orientational specificity, right? Bonding in only one relative orientation, and if you want orientational specificity, you need to asymmetrize the object. Okay, the object must be less symmetric. If it is spherically symmetric, it can form the bond in any orientation, right? So you have to asymmetrize it, which means you're you're basically reducing symmetry. So this globe on the left is an achiral object, right? It doesn't have any. It doesn't have chirality. You can wear it in both hands. Our hands are chiral, and these globes go on both hands. Each of them can go on both hands. But if you asymmetrize it, make the top and the bottom different, these globes are very specific for hands, left and right globes. So so we have asymmetrized them. This is chiral. Okay? And chirality means sorry. So chirality means lack of the reflection symmetry planes. Okay. The question is that why is chiral? Sorry.
You better take this call, otherwise we can't go on.
Guy, yeah. Hello. Sorry, these guys drive me nuts. Sorry.
Yeah, you might want to turn off the phone. Otherwise, I don't think we'll run out of time.
Yeah.
Thanks.
Sorry. So chirality is a problem because when you have racemic mixtures, polymerization does not happen. Okay, when you have a left-handed polymer trying to grow, the right-handed monomers can completely stop the polymerization. So in a racemic mixture, polymerization is impossible. Right? So why did nature come up with this option, the choice of chiral molecules? Chiral molecules are very difficult to polymerize in a racemic mixture. So what happened? What is the advantage for chiral molecules? The advantage obviously is orientational specificity. If you want to bond a molecule with specific orientation, you need a chiral molecule. Particularly for a 3D polymer like DNA, you absolutely need chirality. And that's one of our papers. We provide more pictures, more elaborate in a better fashion. So the evolutionary benefit for chirality seems to come from unidirectional replication. Okay. Otherwise, chirality doesn't make evolutionary sense. Okay. So, so that's the message. Asymmetric cooperativity seems to explain quite a few things of DNA structure, which is strand directionality, unidirectional replication, and antiparallel strand orientation, and heteromolecular base pairing, A and T, G and C, and not A and A, because you don't have any left-right symmetry, asymmetry. And you have 4 nucleotide alphabets, GCSQs, which is what which is observed. Origins of replication, again the same thing. When you analyze the origins of replication of every genome, you will find a flip of GCSQ going from up to down for almost every origin of replication that has been analyzed. And we provide a possible reason for the benefit of chirality. Why is chirality beneficial? Otherwise, chirality doesn't make sense. I'm sorry for the disturbance, but yeah, that's the end of it. And I thank Bob Gedden for giving me the opportunity to do this research and wonderful discussions with IMO faculty and postdocs, and my recent grant for providing me the financial freedom to pursue what I want to pursue. Thank you so much.
Hey, thank you. This is a fantastic presentation. So let's open the floor for questions. I have some questions, but I'll wait.
Surajit, I have a question.
Yeah, Gemini, please.
So the frequency of CG pairs is much higher than the other pairs, right?
It depends on the genome.
I thought in most genomes, one of them, CG or GC pairing, it's much more frequent than any of the other pairings.
You mean dinucleotide frequencies?
Yeah.
Yeah, you're right.
Yeah, is that related to the CG skewness that you were talking about?
Yes, yes, you're right. So yeah, let me go back. Yeah, this one. So you can see that so you can see that if the orange is C and if the blue is G, right? So you've got a CG dinucleotide. And the CG dinucleotide, they each stabilize each other. So this is called a CPG dinucleotide. And it's relatively difficult to break this dinucleotide. So you can, almost in every genome, you will see CPG underrepresented. GC is overrepresented, but CG is underrepresented.
Okay.
So this is possibly why.
Okay. Thanks.
Yeah. Well.
More questions?
I had a question.
Yeah, go ahead. Please.
Okay. You're looking at DNA, but what if you look at RNA? So you obviously won't have the issue of origin of replication, but RNA can exist as a single strand, and it can act as a template to produce a replicate, which is complementary to the single-stranded RNA template. And would things change much in the case of RNA?
The major difference between DNA and RNA is the flexibility of the backbone. RNA is very flexible, which means they can form secondary and tertiary structures in a single-stranded form. So, of course, from an RNA world perspective, that was beneficial. So you can either make yourself as a template and make more of yourself, or you can fold into a tertiary structure and catalyze reactions of your own same type template or other templates in a hypercyclic reaction. But otherwise, the asymmetric cooperativity seems to also work in an RNA type scenario, particularly with the CRISPR. If you know the CRISPR Cas9 system, you can see the interaction between RNA guide, the guide RNA, and the DNA. That is very directional. So when a guide RNA comes and binds with the DNA, it doesn't bind randomly at every point. It usually starts at one point and then zips like that. So it seems to be very directional. There seems to be some asymmetric cooperativity present there as well. So I would, the problem with RNA is that it's, the dsRNA is difficult to make and difficult to unzip with an optical probe, like a molecular, or I mean, a force microscope, atomic force microscope. It's difficult to unzip that. So people haven't done thorough study on dsRNA systems.
Mm-hmm. Okay, I had one more question or clarification rather about the comment you made about the genetic code, the middle nucleotide being key. I didn't quite follow that. So why do you think that the middle nucleotide is the key recognition factor?
Of course, of the three nucleotides, the last one seems to be it's called a wobble pair. So it doesn't determine as much the type of the amino acid that you're gonna code it with, but the central nucleotide dictates basically fundamental properties of the amino acids, like solubility, like polarity, and all that. So it looks like the central nucleotide dictates. So there is a graph, there's a picture that I had, which I eventually deleted. So if you take all these nucleotides with the central pair and group the amino acids just with the central nucleotide alone, and these groups have some specific physical properties, distinct physical properties for each one of those nucleotides.
Right. So I think the main reason is the central nucleotide when you change the central nucleotide, you go down or up a column of the genetic code table. But when you change the first nucleotide, you go across a row of the genetic code table. And
yes,
so along a column, the amino acids which are encoded in the code actually have very similar properties, as you mentioned. But as you go across the rows, which means you are not changing the central nucleotide, but you're changing the first nucleotide, you still have differences in the kind of amino acids that are encoded. But it's true that if you go down the column, you get amino acids, or the encoded amino acids have very similar physicochemical properties. And that's one of the features of the genetic code.
So on a tangential point, so we are basically doing, as we speak, we're trying to unzip. So basically, we are trying to see the triplet code. If you unzip the triplet code with some kind of an asymmetric cooperativity, we are trying to see if the unzipping time correlates, if it groups together, just as the genetic code groups together. Okay, so let's say if you have a four-fold degenerate code, let's say the central for the central nucleotide of A, there is a four-fold degeneracy, right? The wobble base pair doesn't matter. And do they all four group together with the same unzipping time? And if you have the central base pair of CG, something like or G or C or whatever, then you have a two-fold degeneracy. And do you see the two-fold degeneracy in the unzipping time? So that's what we're trying to see right now as we speak. And we see some encouraging signs of that being true.
Yeah, okay, thank you.
Questions?
I have a question.
Who wants to ask, Yuman or Sunny? Sunny, go ahead, and then Yuman. Oops, Sunny, Sunny, did you have a question?
Oh, no.
Okay, didn't. Okay, I'm sorry. Sorry, Yuman, go ahead. I'm sorry.
Yeah, hello, Hema. Can you go to the slide showing the center bubble with the possibility that the center one has more chance to start shifting? Sure. Yeah, sure. This one, the one with the triangle. Yeah, down. To the left bottom. Left bottom. Slide 35.
Okay. Slide, this one?
Slide 25. 35? No, slide. Yeah, 25. Slide.
25. Okay.
Yeah. The one with the triangle. Yeah, this one.
Yeah, this one.
Okay. My question is the following. You have what? 100 sites, right? N equal 100.
Yeah.
At the x-axis. Now, what happen if n is 1,000 or 10,000? Will you still expect the weakest link will be the half of the n?
Okay, so here, just for clarification, here the 100 is just the percentage of coverage. So 100% is full length of the genome. So each genome has a different length. So to normalize the length, they're converting that into a percentage.
Okay, so after you normalize it, then this figure applies no matter how long is a genome.
No. Okay, so prokaryotes are relatively short. You're right. Prokaryotes are relatively short, so you will have one particular V. But if you look at the GCSQ of eukaryotes, which are much longer,
right,
much bigger, you will see many such inverted skews.
Okay, so are you not worried about you form, for example, two bubbles, right? Two bubbles.
Yes.
What happens when they collide and they do not match?
Yeah, so it's just one of the recurring problems. So nobody really has figured out how they decide where to stop. So it seems to be very stochastic. Where these two replication bubbles collide and what happens there seems to be very stochastic. It's still unknown. It's still unknown how these replication machineries stop where they need to stop.
Okay. Since you talk about kinetics, I want to know when you asymmetrically try to unzip the strand of the helix, what is the time scale involved in unzipping the lex pair or forming the lex pair? What is the time scale?
Usually in a second, thousands of base pairs unzip in a second.
So it's kilohertz.
Yeah,
so it's kilohertz.
Yeah, kilohertz, yes.
Okay, then my next question is that does this unzipping per pair depend on the length of the zipped one or unzipped one? Or is it only depend on the next neighborhood? I'm thinking of a random walk, right? A random walk, yes. The Markovian says that you only remember what the last one. Okay. If this is a cooperative unzipping, so shouldn't it depend on a certain length of unzipped or zipped DNA before the next one to be zipped? Is it really a long range zipping?
Yeah. So it's a good question. First, let me qualify that, saying that the unzipping rate, the rate of unzipping, when I say thousands, it's on an average. So the rate of unzipping varies drastically across the genome. And there are places where they're called replication slow zones, where the replication seems to suffer a lot. It just couldn't, so for example, in rRNA, rDNA, where the ribosomal DNA is replicated, where tRNA is replicated, tRNA's gene are replicated, those places are very difficult to replicate. So the replication machinery moves rather slowly. And there are places in like promoters where they move really, really fast. So as you said, there is a possibility. Yeah. Go ahead, please.
So my question is that if you collect this time of unzipping or zipping, what kind of distribution is this time?
Okay, that's a good question. I don't know the answer. I don't know the answer to that.
Okay.
Yeah,
Okay, thank you. Yeah.
More questions?
Yeah, I have one question.
Yeah, go ahead, Scott.
So you had mentioned before that you do not think that there was a enzyme or anything of the nature in early life. So like it's really important, right, that the replication of these nucleotides is able to occur without them. What makes you say this? Like sort of what evidence is there that the origin of nucleotides precedes the origin of enzymes?
So the replication came first, okay, and when you have a template, the information needs to be stored. Information about the enzyme construction needs to be stored in a template. Okay, it has to be self-contained. Your life form has to be self-contained. So the information about enzyme must be embedded within the self-replicating molecule itself. Okay, it cannot come from extraneous sources. It has to be embedded within the enzyme, within the self-replicating template itself.
I see.
So enzyme would come later than replication.
I see. You don't think that there could be other nucleotides that arose simultaneously?
There probably were other molecules that were self-replicating as well, but evolutionarily, these were the ones, the DNA's nucleotides were the ones that won out the race. They won the race because they were superior.
I see. Also, sorry, is it okay if I ask a second question?
Yeah, sure.
Yeah, please.
Do you know if this GC skew holds true in eukaryotes as well?
Absolutely. So if you look at the origins of replication of eukaryotes, right close to the origins, there's a profound asymmetric distribution of nucleotides. And in fact, these days, almost every software that tries to find origins of replication basically does this. They calculate the GC skew and they try to find the switching point.
I see.
whether eukaryotes or prokaryotes, for both.
Interesting. Yeah, because I know finding origins is actually quite difficult in eukaryotes.
Exactly. Yes.
Yeah.
It's been one of the big problems because there is no set motif to look for, like in prokaryotes. Like there is no nothing like a Tata box or CPG islands or something like that. So, but now it's quite regularly they use GC-skew for identification of origins.
And can you then comment on, like, for instance, I know that origins typically are associated with overall having higher AT content, but here you say that it's the CG skew that's more indicative. Is it more indicative or also correlated?
Okay, that's a good point. Sorry, I forgot to make. There is also corresponding AT skew. So AT is also skewed, but that depends on species. How it is skewed depends on the species. So the direction of asymmetric cooperativity depends on different species. So there is AT skew as well. That is also being used. But both these peaks usually coincide. The AT skew and the GC skew peaks, they usually coincide.
I see. Thank you.
Yeah, Scott.
Yes, thanks, Scott. I have a question.
Please, Prashant, go ahead. Thanks, Scott. Scott joined on Newman's invitation. He's an experimentalist, graduate student at Stanford. Great to meet you. Prashant, go ahead. Sorry.
Yeah, Hema, I have a question. See, I'm sort of asking like a physicist, mechanical picture. See, when we have two strands, we have this torsion plus riding, which is a integer number, a self-linking number. So when we are talking about skew, is there any difference in torsion? Torsion is a local property. Because you are being asked about this long range, short range. The riding is like a long range property. Is there anything related to that, for example?
That's a very good question. Apparently, so the torsion is another encoding mechanism of asymmetric cooperativity. So you can have two kinds of torsion, one on this side or one on that side. So it is already asymmetric. Torsion is asymmetric. You can have positive strain or stress, or the other way. So it's already asymmetric. So the asymmetric cooperativity incorporated by the nucleotides can interact with the asymmetry coming from the torsion. And DNA, of course, exhibits many such states. There is something called a zDNA, which is actually left-handed helix. It's not a right-handed helix, it's a left-handed helix. And zDNA have very specific properties. In replication, zDNA has some issues. It's unstable. So there is some kind of an interaction between the torsion and these kind of skews. They both are asymmetric, as you said, yes. Well, on a unrelated note, there are enzymes that can detect if the DNA is highly wound, if the torsion is more, because while you replicate, so you basically wind the DNA in front of you and unwind the DNA behind you, so that the torsion increases, and the topoisomerases come up and cut the DNA and relieves stress. So wherever the torsion increases, whenever the torsion increases beyond a point. And it is amazing to think how these objects can recognize the amount of torsion in the DNA. It's just amazing. I'm not a biologist, but to me, it looks like it has got agency.
Yeah. Thank you. Yeah.
Emma, I wanted to ask you something which is probably a bit too simplistic, but when you look at this problem of zipping, unzipping from the mechanistic point of view that you are, to what degree does the mechanism imply what kind of molecules should play a role? And I ask this question specifically because you showed that the energy barriers play such a critical role. So while it may not implicate one or two or three particular molecules, it should implicate groups of molecules that should play a role such that the architecture can be realized? So I don't know if it's a legitimate question, but that's what
it is. In fact, it is one of the central questions of a field called field of primitive genetic polymers. What people have found is that the AGTC that we use today, the DNA uses today, seems to be very unstable in a primordial scenario. Let's say for cytosine, it doesn't stay as cytosine for too long. It deaminates and it becomes some other molecule. So these were not probably the first molecules used by a genetic system of a protolife. It was probably some other molecule. And they are still, there is still ongoing research about what is this groups of molecules were that led to DNA, that eventually evolved to DNA. So there has to be a chemical continuity between DNA and those primitive molecules. So you should be able to easily make them in a primordial ocean. So there has to be a chemical continuity, but then the molecules have to be simple and they have to be stable in a primordial environment. So what these molecules were, they are still trying to figure that out. And there are various theories about what these molecules were and what kind of environments they were in. One of the popular ones were like black smokers, white smokers, and undersea, deep sea vents. One of that, and another controversial one is the RNAs on ice. So they have a longer lifetime for reactions. And the other one is clay. It has a I'm not sorry, I don't know the name of that clay. It has a very difficult name, but there is a clay. So there are very many options, very many models that have been proposed about what these kind of molecules were.
Oh, that's fascinating to know. I've always been quite interested in the discovery of the, life-related molecules in meteorites and in the asteroids and so on. And the question really is, how primitive are they? Where did they come from? Why they are what they are? And I was just curious, as you were saying, that what is known, maybe it's something to dig around for.
There is also a panspermic seeding. It came from somewhere else because there is a meteorite in Australia called Murchison meteorite.
Murchison
meteorite that showed some amount of amino acids in it. It had some amino acids, and these amino acids were actually chiral. There is some chirality. They were chirally enriched as well. So. So there is a possibility that it was life started off from a panspermic event when our Earth was kind of seeded by meteorites carrying life.
Yeah, right. That's been a thought that's been around for a while, and yeah, fascinating. Any other questions before we close today?
Yeah.
All right, hearing none. Thank you so much, everyone.
Wang, I
want to ask a.
Oh, okay, go ahead.
I want to ask out of the top questions. Do you think the kinetics of protein folding has been understood?
That's a good question. I'm not a biologist, but I do follow some of this. Apparently, the kinetics of the kinetics, at least to some extent, seems to be dictated by the kinetics of translation itself. So while you're constructing your protein, whatever part that comes out first, so you have it's a linear mRNA, and the mRNA gets translated, and whatever translated first, that comes out, and that kind of folds on itself. And then the later part comes and they fold on themselves. So there is this is not captured by the folding algorithms. Okay, because folding algorithms just take the entire sequence and fold it. They do not take into account usually that only part of it first emerges from the ribosome and then folds, and then later ones come and fold. So that folding seem to depend on the kinetics of the construction itself, kinetics of translation itself. So your energy minimum, the folded minimum of the protein, is not exactly a structural minimum, but is also a kinetic minimum. There is some kinetics involved. That seems to be emerging right now as we speak. That's an emerging idea right now, but it jives with what we see in DNA. In DNA, information is encoded in kinetics. With the asymmetric cooperativity, you can think of different sequences having different kinetics. So you can encode information through kinetics, and similar thing can go on in proteins as well.
Yeah, because last year we start looking into this folding problem, and the first thing we have is that people think of this random walk on each amino acid. Then I looked at some video created by someone in University of Washington, Seattle. Okay, and looking at the video, I mean time evolved video. I found that the most important thing seems to be the kinetics of the gamma string, right? Because gamma is a linear one dimension, therefore it can change stochastic very rapidly. Whereas if you look at the beta and the helix, they are very rigid, and therefore they are not going to change very much. So the folding actually should start from the secondary structure, not the primary structure. It's like statistical mechanics, right? We ask, where do you start describing the system? If you start with the amino acid, then you're never able to explain how fast the kinetic folding is. Whereas if you start with the secondary structure, then you can try to describe the kinetics. I think that's what you think, what you tell me just now. You have to start with the secondary structure and try to get the tertiary structure rather than the primary. Therefore, the DNA sequence already coded the one-dimensional thing. And that is easy to understand because that determines the secondary structure, but not the folding. The folding comes from the kinetics of the secondary structure. Yeah.
Yes, I believe so.
Thank you for the. Yeah.
Thank you.
Any other questions? Okay. Hearing none, let me close this. Thank you so much, Hema, and thank you everyone for joining us again.
Thank you.
Really, really a lot of fun and
thank you.
Great learning for the Saturday morning.
Thank you. I'm honored. I'm honored.
Thank
Thanks
you.
for the opportunity. And I'm sorry for the interruption. I'm really
for
sorry.
the interruption. No problem. I mean, you need your packet in the right place.
Yeah, that is important. Yes.
Depending upon the packet, of course.
Thank you.
I don't know what it is.
Thank you. We'll catch up and keep in touch.
Sure, Sunny. I'm sorry I couldn't talk to you initially. Yeah, it's nice to see you after a long time. Yeah, I've been seeing you in other lectures, but I couldn't talk to you. Yeah.
Yeah, Sujay had also joined.
Dr. Sen told me you were here, but you had your cameras off most of the time, so I didn't pay attention. I'm sorry. Yeah.
Sujay is here as well. Hi.
Sujay, I don't know if he's here now. He was.
Okay.
Yeah, Sujay had joined, and Krishna Mohan is here, but I think he's muted.
Okay.
You may remember Krishna Mohan because he was visiting us when you were here.
Okay.
All right.
Nice too.
Wonderful.
Yeah.
Have a great weekend, everyone, and thank you, Scott, for joining us.
Absolutely, it was a pleasure.
Bye-bye.
Thank you.